suspect inside. However, a small trace of blood left behind by the suspect gives policemen some clues. Good afternoon. My name is Aaron Cordova, and this criminal investigation will not be possible without my topic, DNA testing. Friedrich Mischer was the first one who discovered the nuclei of cells. He was born in Basel, Germany, August 13, 1844, and died in August 26, 1895. He discovered this while trying to separate lymphocytes in 1869. During the 1900s, Thomas Hunt Morgan discovered genes in nuclei. He constructed a chromosomes map that consisted of 46 chromosomes and 23 pairs. The purpose of DNA is because back then there were too many crimes and there were too many murders. Also, people were falsely accused of a crime they didn't commit. Let's skip up. Okay, my cards are not in order. Okay, hold on. James Watson, who was 24-year-old and self-trained physicist in 1953, James Watson studied the structure of DNA. He questioned himself how could DNA arrange to hold genetic information and used a process called X-ray crystallography to answer this question. Within cells, DNA is stretched out into chromosomes. X-ray crystallography is a process that James Watson used in order to find the helical structure of DNA. This procedure is close to regular X-ray, but instead of X-ray, you use X-ray radiation. This process is used to crystallize the adenine, cytosine, thymine, and guanine, which are the four nucleic acid bases that make up DNA. Francis Crick, who is 36 and self-trained in chemistry, helped James Watson and his X-ray crystallography come to success. In a double helix, each type of nucleobase strand normally interacts with just one type of nucleobase on the other strand. This is called a complementary base pairing. In a complementary base pairing, purines from hydrogen bonds to pyrimidines, with aguine bonding only to thymine and cytosine bonding only to guanine. In a crime, there's always two sides, the victim and the criminal, which is usually the guilty in a court. When they find evidence surrounding the crime, if it's either a DNA sample, a cheek cell, or a hair root, they use DNA testing. As shown in my create-up, this is an older version of an electron microscope, which is used by scientists when a regular microscope cannot detect the arrangement that DNA can make. In 1779, Alexander Rich discovered the Z form. This form was very different from the regular forms A and B form because it had a zigzag shape, which is why he named it the Z form. This form was the fifth form that DNA molecules can hold to arrange DNA information. In 1983, Adolf Jeffrey was the first one who used DNA fingerprinting while trying to figure out who sexually abused two girls. When policemen found evidence of blood, they used DNA fingerprinting that they found, and 2 weeks later, they found out that the two DNA fingerprintings corresponded to one killer. Then policemen caught the killer. The first step of DNA is the location. The location can be either in a Washington, D.C. laboratory or just as state police headquarters. When there is a crime, there is usually smoke left behind when there's a shooting. Gas chromatography is used when to find out which gun used, like an XL or rifle. First, a sample is ejected with gas into a long coil tube packed with very small grains of a solid. The different chemicals in the sample creep at different speeds through the tube and get to the end at different times. Since each chemical component has a different chemical property that affects the movement through the tube, a sensitive detector attached to the tube measures this, and a computer draws a graph of the results. Each line or peak of the graph shows a different chemical in the sample. The graph where the computer shows the result is called a chemical fingerprinting, because the pattern of the peaks is different for every substance. Step 2 of the DNA testing is fingerprinting. Since each individual contains a unique sequence that is specific to that one organism, this can be used to find the fingerprinting. First, the DNA is cut into small pieces by restriction enzyme. Then a special jelly substance, agarose gel, is poured onto a metal tray. Then electrodes are attached at the ends of the metal tray. Then it is attracted by electrical charge, which will eventually separate the DNA according to its size. Then a nylon membrane is placed on top of the gel, but the DNA fragments remain in order. And then a radioactive dye is called a probe, which is now added to the DNA on the nylon membrane. According to my primary source, Dr. Miller, a piece of X-ray film is sensitive to light, so X-ray film is sensitive to radiation. So then a blob of white and dark patterns will appear, and that's DNA fingerprinting. Step 3 of the DNA testing is the quantification. The quantification of DNA tells scientists the percentage of UV light or ultraviolet light and the amount of human DNA in the sample. First, the sample is loaded in the tray, and then you have fluorescence, which decreases according to the amount of human DNA. Step 4 of the DNA is the amplification of DNA. Amplification of DNA is like cell cloning by making repeated copies of the DNA segments. PCR, or polymerase chain reaction, is a biochemistry and molecular biological technique for enzymatically replicating DNA without using a living organism, just such as E. coli or yeast. The first step is the polymerase chain reaction is heated to a temperature of 94 to 95 to 96 degrees Celsius, or 98 degrees Celsius if extremely thermostable polymers are used. And this temperature is then held for 1 to 9 minutes. This first hold is employed to ensure that most of the DNA is melted by disrupting the hydrogen bonds between complementary bases in two strands. The PCR process is then followed by the annealing step. In this step, the reaction temperature is lowered so that the primers can attach to a single-stranded DNA template. The temperature depends on the diamine of the primers and is usually between 50 to 64 degrees Celsius, or 122 to 147.2 degrees Fahrenheit, for 20 to 40 seconds. The annealing step is followed by the elongation step, during which the DNA polymerase copies the DNA template, starting at the primer's anneal to both of its strands. The temperature at this step depends on the DNA polymerase used. During the extension a temperature of 72 degrees Celsius, or 161.6 degrees Fahrenheit is used. The next step to DNA testing is running the data. This step uses a machine or gel and separates the DNA by size or quantity. The gel acts like a microscopical net. As smaller pieces get through the gel, it separates the DNA by their size. The machine reads the DNA while shooting lasers at the DNA. There are many different kinds of DNA tests. One of them is called forensic, which is used to identify a child to its parents or your identity. Testing on a bug is another DNA test, which is used to find out medical conditions such as cancer or kidney failure. Drug test is used what kind of drug can be used will work on a particular disease. There are many positives to a DNA testing. One of them is that it helps scientists and policemen find out who killed or robbed a certain item, and it helps scientists find the cure for a disease. There are no negatives to DNA testing, but when the DNA gets contaminated by some dirt or bacteria, this can cause a problem. In the future, DNA testing will cost cheaper, since today it costs $2,000 per sample. Then there will be frequent checkups so people can be healthier. They can also check for genetic weaknesses so they can act sooner before in a situation. Thank you for watching my presentation, and as you can see, DNA testing has made our world a better place. I will now take any questions. Mary?
What does DNA stand for?
Your trioxide acid, I think. Yeah. Adrian?
Why is it a problem if the DNA gets contaminated?
It's because when the DNA gets contaminated, you have to get another sample, and you have to redo the whole thing all over again. Araceli?
Is there ever a chance of the DNA being inaccurate?
No. Emilio?
Can DNA be destroyed?
No. Nicole?
How can DNA tests show different diseases?
Well, it shows by detecting some bacteria in the DNA, so they can act for a different drug. Zach?
In the future, you said it'll cost less. Will they still have the same amount of tools to be able to find the research that they do now?
Yes. Garrett?
How does the PCR method, how is it used to test the DNA?
Well, PCR is like making repeated copies of one DNA segment. So it will allow scientists to have more DNA samples. So whenever a DNA sample gets contaminated, they can use that another one.
Okay, one more question.
Julie?
Who performs DNA testing?
Self-trained trained scientists who have studied in college for 2 to 3 years.